Our next speaker is Dr. Islam Hussein, who currently works as a senior scientist for Microbiotics, a company interested in drug discovery. His main research interest includes influenza surveillance and molecular pathogenesis. As a postdoc in the Run Stadler lab at MIT, Islam investigated some molecular markers associated with the polymerase gene of a wild H9N2 isolate of influenza that gained the ability to replicate in human cells after a point mutation. He also studied the receptor binding specificity of a recently isolated H3N8 virus from an unusual mortality event of harbor seals in New England. So hopefully he'll explain all of that to us when he comes up to talk. Dr. Islam Hussein.
So I think everybody here in this room is familiar with influenza as a disease, right? I will try in the next few minutes to introduce you to the virus that causes this disease. This is a picture of the virus. This is how the virus actually looks like. It is very, very small in size, about 100 nanometers. We cannot see it by naked eye. We cannot see it by regular microscopes. And we need a very specialized type of microscopes called electron microscopes. If we dive deep into the virus to unravel its structure, we will find like a rounded virus particle that is surrounded by a lipid envelope, and that envelope carries two important viral glycoprotein. We regularly call them spikes. So I think in blue is a protein called hemagglutinin, and it's abbreviated to HA. And in yellow, we have the neuraminidase, which is abbreviated to NA. Inside the virus particle, we have the viral genome, which is made up of 8 segments of RNA. And this is very, very important for the viral biology, that it is segmented. Influenza virus is not just one virus. It's a very diverse group of viruses. These surface proteins are foreign to our bodies, so our immune system reacts to these proteins by producing antibodies. So we call them antigenic proteins. And according to the antigenic properties of these two proteins, we classify influenza viruses into 18 different HA subtypes and 11 NA subtypes. So each virus will carry one of these glycoproteins plus the 6 internal segments, and we get a garden variety of subtypes that you might hear in the media, H5N1, H3N2, and so on. And as if this diversity was not enough, these viruses keep changing all the time. The polymerase, which is the copying machine that helps the virus to replicate its genome, makes mistakes. And that results in the accumulation of mutations. Here you see the hemagglutinin of H1 virus, and the yellow dots are representing mutations that have accumulated from 1918 all the way to 2009. And of course, this is, by the way, called genetic drift. This doesn't go down very well with our immune system. So for example, if you have antibodies to H1, and these antibodies know that the HA protein looks like a baseball bat, for example. If we assume that after some drifting, the shape of the HA protein transformed into like an arrow shape protein, then the immune system will go, I cannot recognize this, and it will not be able to neutralize the virus. And this is the reason why we need to keep updating our influenza vaccines every year. Another very important mechanism of diversity for influenza viruses is called reassortment or antigenic shift. And this is related to the segmented nature of the viral genome. So if we have 2 virus particles, one in red and one in blue, when they co-infect the same cell, they have a chance of exchanging genomic segments, which results in drastically new, completely new subtypes of influenza that our immune system have never seen before. And by the way, all the past pandemic viruses were generated using this particular mechanism. And all this diversity is exposed to all sorts of selection until the lucky one that carries the right key manages to cross a certain bottleneck or manages to establish a foothold in a new host that could result in an infection of humans, for example, or a transmission from birds to humans. Another important aspect of this virus is that it has a very, very wide host range. It infects birds, and the natural reservoir is aquatic birds. It infects domestic birds, it infects several types of mammals, and of course humans. And this is the reason why it's impossible to eradicate. The cycle of infection usually starts with migratory birds, which show no symptoms of infection. These are the natural reservoirs. And they shed the virus, and sometimes they mix with the domestic chickens, and they get the infection. Inside the chicken body, the virus may evolve into a highly pathogenic form. And of course, domestic birds are very close to us, and that's how humans usually get the influenza infection. And this virus is always on two wings, and migratory birds play important role in the intercontinental transmission of this virus between different countries. For example, Alaska is a very hot spot for transmission between Europe and North America. And very recently, in 2016, we had a transmission of an H5 virus from Europe to North America via Alaska, and it caused a huge outbreak in domestic chickens here in the US that resulted in the killing of more than 48 million chickens. On the chicken, the symptoms are not very characteristic. It can be confused with several other viral infections of chicken. You can see swelling in the limbs, in the head, discoloration. Of course, there will be some respiratory symptoms, but it's very hard to diagnose for sure. In the chicken farm, infection of human can occur through indirect contact. It can occur through touching of contaminated surfaces or through inhalation of droplets or aerosols carrying the virus particles. We have witnessed several spillover events of influenza viruses to humans. The most famous and one of the most dangerous ones is the one that occurred in Hong Kong in 1997. And so, since then, we have also heard maybe you've heard in the media recently about several other spillover events due to other viruses, such as H9N2, H7N9, and H10 and 8, and it just keeps going. Fortunately, these viruses are not yet adapted to humans. So human is like a dead-end host. So they get transmitted from humans to, from birds to humans, and then it does not transmit in between humans. In the unlucky scenario that one of these viruses become adapted to humans, and given our current lifestyle that we travel a lot, this virus can establish pandemic really quickly. If you ask a flu virologist to give you predictions about the next pandemic, they will probably tell you that we cannot predict which virus, we cannot predict when it's going to happen, but it is bound to happen. It's just a matter of time. And that's the reason why we need to get prepared. We need to do surveillance. We need to carry on doing research on vaccines and so on. And that's it. Thanks very much for listening.